Module 7, Exercise Metabolism. So let's look at first an overview of the different fuels that are used during exercise, according to the macronutrient type. So what fuels are used during exercise? Well, for glycogen and glucose, we're going to utilize those substrates for about 35% contribution at rest overall ATP production. If we move up to light to moderate exercise, we get a little bit more contribution from them, about 40%. We go to high intensity sprint-like exercise, we're going to get 95% contribution from glycogen and glucose to ATP production. That tapers down a little bit at high intensity endurance exercise like a marathon. What about fats? Well, fats contribute at rest to 60% of overall ATP production. We move over to light to moderate exercise, we're looking at 55%, 3% for high intensity sprint-like exercise, and 15% contribution to overall ATP production for high intensity endurance exercise. As far as protein is concerned, about 2 to 5% at rest, 2 to 5% contribution for light to moderate exercise, 2% for high-intensity sprint-like exercise, and where we see the greatest impact as far as proteins are concerned to overall ATP production, that's going to be under high-intensity endurance exercise activities, again, like a marathon here, so about 5 to 8%, no more than 10% contribution from protein. Here's just a summary table that shows you, particularly this last column here, the exercise time in minutes that we could get out each of the stored substrates that we have in the human body. And again, this is a model that we've seen before. It's a 70 kilogram male that is 15% body fat and contains about 12% body protein. The exercise time that you're seeing in this last column here, that's a marathon running pace. So if we only utilized one particular substrate as the sole energy source, this last column here tells us the amount of time that we would get out of it. So for example, liver glycogen, we have about 400 kilocalories stored in the liver of energy reserve there. If we performed a marathon running pace, or we were running at a marathon pace, we get about 20 minutes worth of work just out of liver glycogen stores. If we talk about muscle glycogen stores, we'd get 80 minutes. If we simply relied on blood glucose, only 2 minutes. Now take a look at fat. This is kind of interesting. 4,900 minutes that we would possibly get out of running at this pace. However, as we all know, 4,900 minutes simply is not going to happen purely on fat here. And the reason for that is because all the energy systems are important here. So we're relying on glycolysis as well. And eventually individuals hit the wall because of low muscle glycogen stores. So you can have, this 93,000 calories worth of adipose tissue stored, but you're still not going to get your 4,900 minutes worth of exercise time simply because you need glycolysis running at all times. You need muscle glycogen there in order to fuel that particular activity. As far as protein goes, about 1,800 minutes exercise time that we would possibly get out of that. Again, this is a model. This is not real world application. It's just kind of interesting to take a look at the potential energy that each of these substrates have in the human body. So, what is going to determine which fuel substrate we're going to be utilizing during exercise? And the first point that we need to come across is, and understand, is the fact that there is a metabolic demand for oxygen as well as ATP. And when that metabolic demand for O2 and ATP is matched with an equal supply, then we are what we refer to as steady state. So at rest, if we're just sitting in a chair, it's quite easy to generate ATP. It's quite easy to deliver oxygen to muscle tissues, and therefore we're going to be at steady state. Now, we take that application to exercise, and if we have been exercising, let's say for 10 minutes, 15 minutes at the same pace, then there's this metabolic demand for O2 and ATP that the cardiovascular system at that point, as long as the intensity hasn't changed, is being met. So again, we'd be at steady state. Anytime that you deviate away from this, so if you're talking about the onset of exercise, or you suddenly increase the workload, then you get a mismatch in the supply of O2 compared with the demand for O2. So really, this mismatching is going to shift the system that we're going to utilize during exercise for ATP production. So at the onset of exercise or with an increase in workload, the metabolic demand for oxygen, ATP, are going to increase. And of course, since the cardiovascular system has to gear up for this new intensity, there's going to be a lag time before we can actually match the demand for O2 and ATP with an equal supply. So it's all about supply and demand. So over here on the y-axis we have oxygen consumption, or VO2, in liters per minute. It's just a measure of workload or intensity. And on the x-axis we have time in minutes. We have the resting VO2 listed here, and for most people we sit at about 3.5 milliliters of oxygen consumed per kilogram per minute, or what we refer to as 1 met. So 1 met is a resting value. Now if we suddenly begin to exercise here at the onset, we don't automatically go from this resting value to the matching of O2 demand, which is represented by this dashed line that you see here. That doesn't happen instantaneously. There is a lag time there to increase the oxygen consumed. In other words, the cardiovascular system has to have time to adjust in order to deliver sufficient oxygen to the working muscle to match the O2 demand for that particular given intensity. So this oxygen curve ends up looking something like this. So this lag time is represented by this curve, and that's what we refer to as the oxygen deficit. So the oxygen deficit is the amount of oxygen that is needed because of the resulting increase in exercise intensity. So there's a lag time there in order to reach the O2 demand. And once we do match the demand for oxygen, as we've discussed earlier, that's what we refer to as steady state exercise. And for most people, we get there at about 2 to 3 minutes worth of exercise at that given level of intensity level. The minute that you change the intensity, this whole thing starts over again. So just keep that in mind. So at the termination of exercise, again, similarly, we don't just suddenly drop our oxygen delivery to a resting VO2 level if we were going from exercise to rest. There's a lag time there. And this change in oxygen consumption that we see here is what we refer to as the oxygen debt. More newer terminology refers to this as EPOC, or excess post-oxygen consumption. You'll see actually a slow component and a fast component, which is not important as far as this lecture is concerned, but just be aware that you may see that. So, moving on here, as we begin to exercise, we're going to first tap into our muscle ATP and phosphocreatine stores. Why are we tapping into these stores as we change the intensity here? So if we begin the exercise or change the intensity, why are we doing this? Well, ATP is there, it's ready to use. Phosphocreatine, it's there, it's ready to be used. So we tap into those sources. The reason why we start to tap into creatine phosphate refers back to the fact that when we break apart ATP, we end up with ADP, which actually stimulates creatine kinase. And creatine kinase is the enzyme that actually breaks apart creatine phosphate stores. So if you start thinking about the metabolites of metabolism, so ADP is a metabolite of ATP breaking apart, the hydrolysis of ATP. And you start thinking about, well, if I increase the concentration of ADP in the muscle, well, that's going to stimulate things. And in this case, it's stimulating creatine kinase, which then tells the body to break apart creatine phosphate. We also, at the same time, start to get a little bit of contribution from non-oxidative glycolysis. So that starts contributing to ATP production. Again, ask yourself the question, why is this happening? The reason is, well, because if we're breaking apart ATP, then we've got ADP in higher concentrations as well as higher concentrations of that phosphate that's been released. That will stimulate phosphofructokinase, PFK, which is a key regulating enzyme of glycolysis. It's the point at which we go from fructose 6-phosphate to fructose 1,6-diphosphate. And then that point, if you recall, in the glycolytic pathways is where you're going to get a splitting of glycolysis into two separate pathways. So this enzyme is a key regulating step. It's so important to controlling glycolysis and the rate at which glycolysis will operate at. So in this instance, since we've begun to exercise, we're going to increase the amount of ADP and inorganic phosphate, and that stimulates PFK. So that's basically going to rev up glycolysis a little bit. These same metabolites, ADP and inorganic phosphate, are also going to stimulate another enzyme, the branched-chain oxoacid dehydrogenase, or BCOAD for short. This is a very important enzyme that's going to regulate the use of branched-chain amino acids, which are a part of the glucose-alanine cycle, which I've got a diagram here on the next slide that I'm going to show you. So if you recall, we do utilize amino acids for ATP production at rest from the branched-chain amino acids. That represents about a 5 to 8% activity. During exercise, branched-chain amino acids actually can increase quite a bit, and we get a 20 to 25% increase in activity in the glucose-alanine cycle. Now, it's important to note that if you have a decrease in muscle pH, why would we have a decrease in muscle pH? Well, we have the production of lactic acid, which shifts the pH downward and more acidic. So that is going to contribute here to a stimulation of branched-chain oxoacid dehydrogenase. Glycogen depletion also does the same. And if you think about this, if we have glycogen levels that are being used up and we have lowering and lowering levels of glycogen, we need to have a backup mechanism to keep producing ATP. And so the branched-chain oxoacid dehydrogenase is going to gear up the whole glucose-alanine cycle so that we can produce ATP. So let's take a look at the glucose-alanine cycle. We'll take it step by step here. So first off, in the bottom right-hand corner of your screen, we have glucose to pyruvate. We've seen that before. That's glycolysis. The next step here, the conversion of pyruvate into alanine. Alanine is an amino acid, is very, very important as far as the glucose-alanine cycle is concerned. And so how does this happen? Well, if we go over here to the left-hand side of the screen, and by the way, this is in muscle tissue. So in muscle tissue, the BCOAD, that enzyme that we just talked about, is going to be increased as a result of having ADP, inorganic phosphate, the lowering of pH, depletion of glycogen. All of these things are going to contribute to increasing or stimulating BCOAD. What's going to happen there is that we start utilizing branched-chain keto acids in the citric acid cycle or Krebs cycle or TCA cycle. And the way that we do that is we start utilizing our branched-chain amino acids, so that's leucine, isoleucine, and valine. And via the branched-chain amino acid aminotransferase, we take these branched-chain amino acids, convert them into branched-chain keto acids by basically interacting with alpha-ketoglutarate. And if you recall, alpha-ketoglutarate comes from the citric acid cycle. So that's already there, available, and we're going to utilize it in this case so that we can take branched-chain amino acids, combine it with alpha-ketoglutarate, and then we end up with glutamate, which then will interact with pyruvate to allow us to produce alanine, this amino acid. So this alanine then gets shipped out into the bloodstream, and eventually the liver receptors filter alanine out, and in the liver, so this top portion of the diagram here, this is the liver. In the liver, the alanine is then going to be converted back into pyruvate, again interacting with alpha-ketoglutarate, and leaving glutamate behind. And that pyruvate then can be converted back into glucose, which can be shipped back out into the bloodstream and eventually come back into the muscle. So it's a bit of a recycling process. The only catch here is up in the upper left-hand corner of the screen, and that is when we have glutamate now. This is an amino acid, so we have to deaminate this amino acid. So that means stripping off that amine group or that nitrogen group. The liver will take this and run the urea cycle, produce some urea, and that's going to be sent out into the bloodstream and eventually get filtered out by the kidneys. So that's the glucose-alanine cycle. So at the same time that all of this is happening, we also have calcium that's being released for muscle contraction. If you recall your physiology lessons as far as skeletal muscle and how they work, we need calcium there to trigger the sarcomeres shortening. So that's going to be released. And the interesting thing is that calcium is going to stimulate PFK. So again, this key regulating enzyme in glycolysis is going to be stimulated by calcium so that you can increase the glycolytic processes. That same calcium is also going to stimulate phosphorylase kinase, which phosphorylase kinase converts phosphorylase B into phosphorylase A. Now, this enzyme is very, very important in regulating glycogenolysis. That is, breaking apart glycogen in muscle so that you release glucomolecules that you can utilize in glycolysis. So that's a very, very important enzyme. And the beauty of this is that as calcium is being released because of muscular contraction, you're ensuring that there's going to be glucose available by stimulating glycogenolysis. Now, this same calcium, as we have seen before, is also going to stimulate GLUT4 translocation from the intracellular compartments to the plasma membrane of that cell. And of course, GLUT4 transport protein is going to drag glucose into the cell from the bloodstream. And so this again helps to ensure that we've got plenty of glucose available to run glycolysis when we need it. And again, remember, please don't lose sight of the fact that what we're talking about here is that we've increased the metabolic demand for O2, and so we've increased the level of intensity, whether that be from rest to an exercising level, or we've bumped up the exercise intensity from a lower level to a higher level. Okay, so as all this is going on, we also have something called the lactate shuttle, or gluconeogenesis. The lactate shuttle, it's also known as the Cori cycle, by the way, is an important process here that goes on and will take lactic acid that's being produced by the muscle and actually recycle that lactic acid. Because again, lactic acid will lower the pH of muscle, it'll lower the pH of blood, and so we want to try and mitigate those changes in pH by doing something with that acid. So what happens here is that the lactic acid that's produced in muscle is going to be transformed into lactate, which is just a salt version of lactic acid. That's going to be released into the bloodstream, and eventually the liver receptors will take up that lactate, and we're going to transform that lactate into glucose 6-phosphate. And remember, that glucose 6-phosphate is part of glycolysis. It's one of the steps that we've seen. So we basically then can take that glucose 6-phosphate, convert it into glucose, and then release it back into the bloodstream. And eventually that same lactate that we saw that was converted into glucose, that glucose now can return back to the muscle tissue so that we can utilize it. So it's a nice way of recycling lactic acid. Now, there are some special notes about lactic acid that are important to discuss here. The first of which being that the fact that lactate needs some sort of way or mechanism to get out of muscle tissue. And the way that it does that is via facilitated diffusion. That means that it's got a buddy to take it out of the muscle cell. And so it's going to use the monocarboxylate transporters, or MCTs. These are oftentimes in the literature studied in relation to lactate and exercise. So that may be something that you come across in the literature. The next point is that the heart actually loves lactate. It will utilize lactate and draw it from the bloodstream to basically oxidize it and make ATP. And of course, this point here is very, very important, because oftentimes lactic acid is associated with this negative aspect of exercising. And what I'd like to impress upon everyone is the fact that it's actually a very, very necessary entity in keeping activity going, keeping glycolysis going, because it's going to regenerate NAD. When we form, or when we run glycolysis and we make pyruvate, we're actually tying up these NADs with electrons or hydrogen ions. So if you recall, the point of the electron transport chain was to take all the NADH2s and the FADH2s that we've collected and to utilize them in the electron transport chain to make ATP. Well, you know, glycolysis only has a certain number of NAD hanging around, and if you tie them all up, you'd shut down glycolysis, as well as you'd get a buildup of pyruvate. That would also shut down glycolysis. So in order to keep glycolysis going, we very cleverly take pyruvate, strip off the two hydrogen ions that are on NAD, tack them on the pyruvate, and we form lactic acid. And so by doing that, we make more NAD available so that we can keep glycolysis running. So that's a really, really important point, that lactic acid is an important part of exercise. Now, of course, at high levels of lactate in the blood, that will cause a decrease in exercise performance. So there's a point of diminishing return here. So at the initiation of exercise, at bumping up the intensity of exercise, lactic acid formation is very, very important. If we produce a huge amount of lactic acid, then of course we're going to see some decrements in performance. So there's only so much that's going to help us. And finally here, the last point, each lactate molecule is actually going to generate about 14 to 15 ATP. So there's a pretty decent amount of ATP that's going to be produced. And the varying level here, 14 to 15, is basically dependent upon whether you use NADH or FADH in order to produce that ATP. Okay, so in the midst of all this that's going on that we've just discussed, we also start to warm up, so to speak, oxidative glycolysis, the citric acid cycle. Okay, why is this happening? Well, again, let's go back to answering the question of what's available as far as metabolites are concerned. We've got ADP concentration increasing, we've got calcium concentration increasing, and we have NAD that's available. That's all going to stimulate an enzyme in the citric acid cycle called isocitrate dehydrogenase. And that enzyme is located right here. It's the step that goes from isocitrate to alpha-ketoglutarate. So that enzyme, if you stimulate that enzyme, you're going to rev up glycolysis. So again, think about what's being released as a result of increasing exercise activity. We've got a lot of ADP hanging around. Calcium is being released because of the muscular contractions, and NAD can also stimulate this. So we also have to have a way to not only increase the citric acid cycle, but we also have to do the same for the electron transport chain. So again, why are we increasing the electron transport chain? Well, we want to make ATP, and the reason why this happens is because we have lots of ADP and inorganic phosphate hanging around. That concentration is increasing. That's telling the system that we're breaking apart ATP at a pretty high rate. And so that will cause the stimulation of cytochrome oxidase, which is located in the electron transport chain. So you increase this enzyme, then you're going to start to rev up the electron transport chain some more. Okay, so as the metabolic demand for ATP and oxygen approach equilibrium, so in other words, we're getting a matched supply of oxygen for the given demand, then oxidative metabolism is basically going to take over as the primary fuel substrate usage to produce ATP. So we're going to make ATP using oxidative phosphorylation or oxidative metabolism in order to meet the demands, the given demands. So the cardiovascular system under this scenario has delivered sufficient amounts of oxygen to the working muscle for the given demand that's being placed on the muscle. Okay, so there has to be some sort of metabolite here, which we're going to talk about, that triggers all of this. And so ask yourself the question, why? Why is this happening? Well, we get an accumulation of oxygen in the working muscle. We also build up citrate, which is in the citric acid cycle. ATP starts to accumulate a little bit. And we also get some accumulation of creatine phosphate. So those substances, if you start to get an accumulation of them, will actually start to slow down PFK. They will inhibit PFK. If we inhibit PFK, we slow glycolysis down as we're increasing the oxidative pathways. We'll also get an accumulation of glucose 6-phosphate, which means that you're going to slow down hexokinase activity. So you won't draw as much glucose from the bloodstream into the working muscle because this glucose 6-phosphate has accumulated. That's telling the system that, hey, we're matching the demand for ATP here. We've got it. Oxidative metabolism is covering that demand. We also have these substances that are slowing down glycolysis, which will allow the citric acid cycle and the electron transport chain to take over as the ATP producers. So at the same time, again, that all this is happening, we have epinephrine being released, which into about 5 to 10 minutes of steady state exercise, you'll see a higher concentration of epinephrine. So epinephrine is an important player in exercise. Why? Because it's going to stimulate the release of free fatty acids from triglyceride in adipose tissue and muscle tissue. Now that's important because if we want to utilize the oxidative pathways, then it behooves us to utilize fatty acids. We get more bang for our buck that way as far as ATP is concerned if we have free fatty acids available. And so this epinephrine release is actually going to stimulate the hormone, which is called hormone-sensitive lipase. It's very similar to lipoprotein lipase, except that it responds to the release of epinephrine. And what's going to happen here? Well, we're going to take that triglyceride that's stored in adipose tissue and we're going to break it apart into its constituents. So glycerol, backbone, and the 3 fatty acids. So that glycerol is going to be transported to the liver. We're going to turn that glycerol into glucose. So we're going to perform gluconeogenesis. And then the free fatty acids are actually going to be transported in the blood by the protein albumin, the visceral protein, which will then carry it to muscle tissue. And that albumin can carry about 10 fatty acids. So there's a significant amount of fatty acids that are going to be delivered to the working muscle. So we can take advantage of oxidative metabolism. So all of this is going on at the same time. So if you see the picture here, we're starting to, we kick up initially glycolysis, but we start warming up the oxidative pathways, and then eventually when we have the oxygen demand met, and the ATP demand is being met for that given level of activity, then we start to shift over more to oxidative metabolism. So let's take a look at basic skeletal muscle structure, and there's really one thing that I want to point out so that we orient ourselves to where this fatty oxidation is occurring. If we look down here, the bottom right-hand corner of the screen, we see the sarcolemma, or the sarcolemmal membrane, which sits below the endomysium layer. So it's this clear layer that you see here that surrounds these myofibrils. This is where the sarcomeres are at, in the myofibrils. So the sarcolemmal membrane is basically where all of this fatty acid oxidation that we're going to talk about as far as transport is occurring at. So, free fatty acids have been released into the bloodstream as a result of epinephrine releasing or performing hydrolysis via hormone-sensitive lipase of adipose tissue triglyceride. So we've got these free fatty acids floating around in the bloodstream. They need to be taken up into the muscle, and they do so via that sarcolemmal membrane that we just saw. And there are transport proteins, FABP and FAT/CD36 transport proteins, and they're going to be a part of facilitated diffusion. And so they drag in that free fatty acid across this sarcolemmal membrane. They are also going to be activated by tacking on a coenzyme A to form this acetyl-CoA free fatty acid complex. So once we have them in the sarcolemmal membrane, then we're going to activate them by tacking on this CoA so that we can internalize them across the outer mitochondrial membrane and into the inner mitochondrial membrane. So acyl-CoA synthase is what's going to catalyze this entire reaction. And the bad news here is that we take up 2 ATP in order to activate this, but in the long term, we're going to get a lot of ATP, as we saw in the previous lecture, from utilizing free fatty acids for energy. So this coenzyme complex of free fatty acids is going to transport across the outer mitochondrial membrane, and then that's going to interact with carnitine, which is going to bring in the long-chain fatty acids. So you actually just leave behind the CoA, and that's going to take it into the inner mitochondrial membrane, so then you can perform beta oxidation. Now you should note that short-chain and medium-chain fatty acids, they can actually diffuse across the mitochondrial matrix without the presence of carnitine or without the use of carnitine. So that's a benefit to short-chain and medium-chain fatty acids. The long-chain fatty acids always present a problem as far as transportation are concerned. These short-chain fatty acids and medium-chain fatty acids also oxidize a bit more quickly because they are smaller in carbon length, and they do oxidize more completely than long-chain fatty acids. So here's a diagram to put all of that rhetoric into perspective. So let's start at the top left-hand corner of your screen. And what do we have here? We have this circle that represents albumin with some fatty acids tagged onto it. And as we talked about, we want to get those fatty acids across the sarcolemmal membrane in muscle. So we have the FAB/FAT CD36 transport protein that's going to drag that fatty acids or those fatty acids across the sarcolemmal membrane. Then you're going to have fatty acid binding protein going to further transport those fatty acids to the outer mitochondrial membrane, and as we saw earlier, we'll take some ATP to activate this thing, but we're going to tack on a CoA to the fatty acid to make this acyl-CoA fatty acid complex, and that is done by acyl-CoA synthase, the enzyme. So that's regulating this whole reaction rate here. And so this fatty acid acyl-CoA complex then, in order to be oxidized, needs to combine with carnitine here, and carnitine via carnitine acyltransferase 1 will then take that fatty acid complex and turn it into fatty acylcarnitine, which then we release the carnitine and we tack on another CoA to this via carnitine acyltransferase 2, which then leaves us with fatty acyl-CoA in the mitochondrial matrix. So we've transported these long-chain fatty acids via a series of membranes. The sarcolemmal membrane, the outer mitochondrial membrane, and then the inner mitochondrial membrane. So there are a lot of different enzymes, a lot of different steps here in order to get that fatty acid, that long-chain fatty acid, into the mitochondrial matrix so that we then can perform beta oxidation and take advantage of all those acyl units, those acetyl-CoA units, acetyl units, which are 2 carbon units, and utilize them in the citric acid cycle for ATP production. Now, there's one note that I just want to back up and also discuss here that is important, and that is at the level of the fatty acyl CoA, if we weren't talking about exercising conditions and we were talking about storage of fatty acids in muscle tissue, then we'd actually take this fatty acyl CoA and store it. We take some glycerol, alpha glycerol phosphate, and we can have some storage of that fatty acid as intramuscular triglyceride. So, remember that what we're talking about, that was all generated by epinephrine release. So epinephrine released those fatty acids into the bloodstream, and then they were transported into the muscle tissue. But that same epinephrine is also going to stimulate phosphorylase kinase. And if you recall, phosphorylase kinase is going to stimulate glycogenolysis. So we're going to convert phosphorylase B into phosphorylase A via phosphorylase kinase, which then is going to cause the release of glucose from glycogen. So if you think about this, you've got another primer here that's kind of acting as a backup mechanism to release more glucose into the system so that you can take advantage of glucose in glycolysis to produce ATP. So you get not only from epinephrine the stimulation of free fatty acid release, but you're also getting glucose release from glycogen. So that's all ensuring that we have plenty of substrate available to produce ATP given the activity that we're performing. So what's happening hormonally and overall as far as all of these different processes? Well, as blood glucose concentration drops with exercise and sustained exercise, we start to see an increase in glucagon from the alpha cells in the pancreas. And of course, glucagon, when we have glucagon floating around in higher concentrations, it's going to promote breakdown of nutrients, of stored nutrients. And that's exactly what's happening during exercise. We're tapping into the resources that we worked really hard to put away for times of need. And so that glucagon is going to stimulate gluconeogenesis in the liver. And we can utilize the pyruvate that's in the liver. We can utilize the lactate that we got from the bloodstream from that working muscle. We can utilize the glycerol that's being released from the stored triglyceride in adipose tissue. And we can also take advantage of amino acids, like the branched chain amino acids. Glucagon is also going to stimulate glycogenolysis, so it's going to further facilitate the breaking apart of glycogen to release those glucose molecules. The main goal here during exercise, because of glucagon being released, is to simply try and maintain a decent glucose concentration in the bloodstream. Now, why do we need this glucose concentration to be maintained? Well, it comes down to the central nervous system. Central nervous system has a requirement for glucose, as well as erythrocytes or red blood cells. So you always have to have some kind of glucose available in order to maintain proper functioning of those cells. And the type of exercise actually is going to influence this as well. Lighter exercise doesn't seem to have as much fluctuation in glucose concentrations as does moderate to high intensity exercise does. So if you're wondering, by the way, what high-intensity exercise or what moderate exercise is, a MET level of greater than 6 METs is going to be considered high-intensity exercise. So 3 to 6 METs is the moderate intensity level. Okay, so let's look at all of this put together graphically. So in these series of graphs here, on the y-axis, we have glucose on this panel, we have insulin on this y-axis, we have glucagon on this y-axis, and we have free fatty acids on this y-axis, and epinephrine on this y-axis. On the x-axis for all of these figures, we're looking at exercise out to about 90 minutes in time. So we have time on the x-axis. Now you're going to see 2 lines for each of these graphs. The green line represents an unconditioned individual who attempts to exercise, and the red line will represent athletes who are well conditioned. So let's see what happens here. So as far as glucose concentrations in the unconditioned individual compared with a conditioned individual, you can see that the conditioned individual is able to better maintain glucose levels in the bloodstream. And that really spells the fact that these individuals who have conditioned their bodies can rely on other sources, i.e. muscle glycogen and intramuscular triglyceride, rather than relying on the bloodstream for glucose. So as far as insulin is concerned, insulin levels are going to be a little bit higher in the conditioned individual. And again, this means that we're not relying as much on glucose in the bloodstream, and we're relying on other stores so we can maintain our insulin levels a little bit better. As far as glucagon is concerned, again, the conditioned individual has a lower level of glucagon in the blood. As far as epinephrine is concerned, epinephrine differences are quite substantial. The conditioned individual, again, does not have as much epinephrine release. And as far as free fatty acids, which goes hand in hand with this epinephrine, again, the conditioned individual is going to have a higher concentration of free fatty acids, and that's simply because they're more conditioned. They've have the mechanisms and the enzymes in place in higher amounts to release more free fatty acids for a given level of epinephrine. And so you have more availability of this substrate. You've got more stored in muscle as far as triglycerides are concerned, so you rely less on substances from the blood. So that's just a nice summary of everything that we discussed previously, just graphically speaking. So to put these diagrams into some rhetoric here, some summary of everything that's going on, everything we discussed. So the end result here is that we're getting glycogenolysis rate is going to increase. So the breakdown of glycogen is going to be enhanced during exercise. Glycolysis is going to run at a much higher rate. Oxidative phosphorylation is going to increase, so we're going to rev up the citric acid cycle and the electron transport chain more. We're going to produce more new glucose from gluconeogenesis in the liver. And we're also going to increase lipolysis. So all of these systems are increasing just because the demand for O2 and ATP has changed. You've increased the demand, now there has to be a matching of this demand with an equal supply of O2 and ATP for that given level of exercise intensity. This slide shows you a summary of all of the substances that we talked about, basically the control points of exercise metabolism and whether or not they inhibit or stimulate and then the end result. So let me just walk you guys through this. So here in this column we've got the substance. All right, so whether we're talking about ADP and inorganic phosphate, calcium, NAD, etc. This column here is a column that represents, well, if you've got, for example, oxygen, that's actually going to inhibit phosphofructokinase, or PFK. In this column, we have all the substances that are stimulated by those particular substances in column 1. So, for example, ADP is going to stimulate creatine kinase and isocitrate dehydrogenase. And the last column is the end result of this, either inhibition or stimulation of that substance. So if we just take ADP as the example, so we said that that was stimulating creatine kinase and isocitrate dehydrogenase. Well, if you stimulate creatine kinase, that's going to cause more release of inorganic phosphate from creatine. If you stimulate isocitrate dehydrogenase, that's going to increase the citric acid cycle. So I would go ahead and take a look at each of these and relate that back to the big picture of what's going on here. Again, this is all about trying to match the increased demand for oxygen and ATP during a given level of exercise intensity, and this shows you all the little pieces that are going on at the same time in order to make that possible. Okay, so I want to talk about the crossover point in exercise intensity. And this is an important point to understand because there is a shifting of fuel sources with exercise intensity, as we have just seen with all of the control points. And so I just want to relate this back to macronutrients, the macronutrients, carbohydrate, and fat. So what's happening here? Well, we're going to shift as exercise intensity increases. We'll shift over from utilizing fats to more carbohydrates. Now, why is this happening? Well, as exercise intensity increases, we're increasing the recruitment of those fast motor units, or those fast muscle fibers that rely more so on carbohydrate sources, so on glycogen. We also have an increase in blood levels of epinephrine, so remember that that's also causing glycogenolysis in muscle, as well as the release of free fatty acids. Fats, in comparison with carbohydrates, actually are much slower to be delivered and to break them apart, to disassemble them, as well as beta oxidation is slower to produce ATP. And the downside here of increasing exercise intensity is that, of course, that oxygen demand, you need to have plenty of oxygen around. And at higher intensities, you may not necessarily be able to keep up with the oxygen demand. So we rely more so on carbohydrates at the higher intensity levels, generally speaking. So let me show this to you by using this lower diagram here in the lower right hand corner. And so this diagram is showing you on the y-axis the percent energy from fat and carbohydrates here. And on the x-axis, we have the percentage of VO2 max. So VO2 max being the max amount of oxygen that an athlete can consume, which would relate to the highest level that they can perform at and utilize oxygen efficiently. So the blue line represents the carbohydrates, the red line represents fats. The crossover point is right here, and that's happening in this diagram at about 35 to 38% of VO2 max, roughly speaking. And that crossover point, again, just points to the fact that carbohydrate usage is going to be increased at these higher intensities, whereas fat utilization is decreasing with exercise intensity. It's not that we turn off fat oxidation and turn on carbohydrate oxidation. It's just that the contribution from fat for ATP production is going to slow down as that exercise intensity increases. We also need to talk about the delay time and the maximal rates of fuel utilization. And that word is very, very important, maximal rate. When we start to exercise, we start to kick in fat oxidation at about 20 minutes, but we won't see maximal fat oxidation till we're out at 2 hours and beyond. So it takes a while for all of these systems to really gear up and contribute to being the primary ATP producers. As far as glucose from the blood oxidation and utilizing that, at about 90 minutes we start to utilize a lot of glucose from the bloodstream to help us produce ATP. As far as glycogen oxidation, so muscle glycogen, that's happening very, very quickly, 1 to 3 minutes, and we're utilizing muscle glycogen. As far as glycolysis, man, it's almost instantaneous. The second that we start walking or start running, whatever activity that we're doing, we're kicking glycolysis up to the next level, and it's contributing to ATP production. So within about 5 to 10 seconds. Same thing with lactate production. So the minute that we start producing more levels of lactic acid, it's being converted into lactate, and that lactate shuttle is put into play so that we can recycle it and make ATP out of it. As far as phosphocreatine breakdown, that's instantaneous. That's happening the second that we have any change in the concentration of ADP in inorganic phosphate. This last diagram is kind of nice because it puts together a whole bunch of different activities and types of sport on one graph, and it attempts to very rudimentarily, I might add, break things apart into the percentage that it's aerobic versus the percent that it's anaerobic. So let's just take a look at the extremes so that we can understand this a little bit better. Here, if we're looking at activities that are 0% aerobic, so they're high intensity, they're short duration, diving would fall into that category, gymnastics, if we go out a little bit more to almost 10%, and football. So those are very, very high intensity activities. They're of short duration, not as far as overall like football game-wise, but as far as utilization of fuels for a particular movement in that sport. If we look at the other end of the spectrum and 100% aerobic activity, we're looking at the jogging being 100% aerobic, and something like a marathon as well, being out at that end of the spectrum. So I would take a look at this diagram and think about these types of sports and relate that back to the control points that are basically determining what system we're going to utilize primarily as the primary contributor to ATP production. So is it going to be glycolysis? Are we emphasizing more glycolysis, or are we going to emphasize more oxidative metabolism, that citric acid cycle and the electron transport chain? So the final point here to remember is that we never turn any of these systems off. It's just we're increasing one and decreasing the other, kind of like a seesaw going back and forth. We emphasize one at one point, and then we de-emphasize it and emphasize the other system. So it's going back and forth to match that O2 demand and that ATP demand. So it's all about that demand and how we can bring ourselves to steady state.